that telling species apart by eyes are really difficult. Even the books and all that, like, okay, is that, oh God, is that a, what? No, like you read the keys, it's just like, yeah, I've been there, and not just been there, I'm in there. I like, that's my life every day, and have to like figure out that species from the others. So like, for example, like this is a like the book I wrote for my class, and this all one tree look nice, all mossy. I tell you right now, there's like 5 species on them, and you're like, what? There's 5 on them? Like, how could you tell? Like, I don't know most of the time. So some group, like even critic species, are really difficult to tell, and sometimes we just want to know the species are like, not all people, not all biologists have the same patience that I do, right? Some people just like, I just want to study this particular species. Just tell me what it is. And you get so many of those people. In case you don't know, or you probably don't know, I man the Facebook page for my department, and my department is Department of Botany. So we got a lot of this people sending pictures. What is this tree? What is these flowers? Like, where do we find this tree? It's like, I don't know. I'm not a taxonomist. I don't know. I don't know. I don't know big plants. I work with small plants, and I can't tell them that because I'm the admin of the page. So I would send the picture to the expert in my department, and they were like, "We don't know either." It's like, oops. So, yeah, exactly. But the thing is, like when you take picture of plants, and most people have this crazy idea that like, oh, you take a picture of any plants and you can tell right away what plant is. The truth is, like, let me tell you right now, if you're gonna do that to my Facebook page, take the picture of the flowers. We cannot tell things from the leaf, people. Sorry. Seriously, like leaf are great, but they're not definitive of the species of the flowering plants. You need the flowers. You need the flowers. So we most of the time we'd be like, yeah, well, you think Google can do that? Not yet. Let me tell you right now, not yet. You would hope so, but not yet. I guarantee you that we like it's go back to the first problem that we currently not properly delimit our species yet. So even we give Google the input, they don't have a proper input to tell us what it is. What if it isn't angiosperm? Even worse, because if you study ferns, you need to look under their leaf and take picture of the sporangiums. And you with moss, you need a sporophyte, and it's like specific. Each group have like specific thing they need to see. And for me, like if you give me a moss, you can give me everything you have on that moss, and I still can't tell you what it is. So that's just how difficult things are. So we do this. So like let's say we know this what the species are for sure, but then like what if we forget about opening all books or relying on smart people at Google to do image analysis for us to figure out what it is? Let's say we do it going to the core of the species, like let's use DNA to figure out. We talk about DNA testing all the time, right? But we talk about in the context of like paternal testing, or like who's father, who's a murderer. But if we use DNA as a way to tell the species, just like barcoding, you go to the convenience store, right? And they will like, beep, and they know exactly how much is that thing and when it's produced, where it come from, what kind of product it is. It's the same thing. What if we can use DNA as a barcode to identify the species? What if we can do that? Wouldn't that be nice? Just like you don't have to like invest a lot of money to like analyze all the images. It's just like, can I get a little bit of your blood and sample your DNA and like beep, well done, already known. So wouldn't that be nice as a future, right? So the same idea here, because all the organism in the world, include like all the way to prokaryote, with exception for some viruses, they all have DNA. So DNA is at the core of all the cells, and there must be some area that provide a barcoding or signature of that particular species. If I look at that gene and like, that gene have this signature, it belongs to that species. And if you look here, you can see that these two butterflies are the same species, but they come from different countries. The barcode like looking thing are pretty similar. So you can think of ATCG as just like different strip of barcode. The barcode only use like the black and white to tell the space, right? You use this one be better. We have four. We have ATCGs and keep and the position and the abundance of each base, you can tell apart. And you look at these two different owls, they two different genera, and their barcode will look different. So the idea is really simple: is that find that region that tell us what this species is. So that's the job of DNA barcoding. And so there's three steps to DNA barcoding. So first you extract DNA. Second, you amplify the barcode regions. You do the PCR. So this is a PCR step. Very simple. And then once you get that region, you compare it against the database. So just like you go to 7-Eleven, you take the product out, right, which is DNA. But in this case, your DNA probably too tiny. You have to amplify them, and you only rip out just the barcode part, not the whole thing. And then you show this barcode to the system, like, what is this thing? Like, this might be a frog, like three-legged frogs. Like, okay, sure, right? There's no such thing as three-legged frogs, but there's a disabled one, but yeah, we should not bully him for that. So that's the three step. It's really simple. That's the idea. And this is exciting because why? Because DNA for barcoding can be from anything. It can come from fresh specimen, come from herbarium specimen, can come from environmental trace, basically like water, soil, anything. It's to the point that like everyone, like let's say you in the room, and by the time you leave that room, you already leave some of your DNA in your like skin cells or whatnot. And the janitor or the housekeeper can come in and sweep everything, and they can put this into the machine and can tell us exactly who will be in this room. They can do that. They could do that in theory. So you have a lot of money and you have free time, want to figure out who's in this room without actually looking at the cameras, and they don't require the whole organism. And this is really exciting for people who study ecology, because you can go into the water, right? You want to see in this water who's live here, and you just sample all the water, and then you're done. Like you know exactly who's been here, and this will take a lot. And the cutest thing I see, I lost the video, but they go into deep jungle of Amazon, and they found these tiny little frogs, and they're so tiny. It's like, oh yeah, this is really rare. It's like 100 of this in the whole wide world. It's like, whoa, they're tiny. And if you plant, people be like, oh yeah, just break the legs and crush them in DNAs, and like you can't do that with animals, at least not ethically. They used to do that in the past. So back in the day, if you're studying frog DNA, you have to like clip their toes a little bit, like one of those webby thing, just clip them just tiny bit, like just tiny little bit to get the DNA. But with this new method, you don't have to do that anymore. You just come in with a cotton swab, like small cotton swab, and just like stroke at the back gently. It's like, oh, poor little thing, can I get some of your DNA? Those mucus have some of DNA, and you put that into the tube, and you can sequence DNA from that. So that's just how simple it is. So it doesn't take a whole lot of materials, and you can work with very old specimen, and people really go crazy as they did extract DNA from Neanderthals. So we now know what Neanderthals DNA looked like and how much of it remained in us. So our naughty ancestor, Homo sapiens sapien, did mate with a Neanderthal. So all of us have some Neanderthals in our blood. So good to know. So this step is easy, and Ajahn Pim can tell you all about this, about DNA, biochemistry, which I won't gonna bore you with that. And then the next part is also pretty good. We have a pretty big database, and there's a few of them. There's like bowl system, there's GenBank. GenBank is one of them. And you can basically get your data and input that into the database. And you probably learned that from um, um, bioinformatics. You can do blast search to compare your sequence with the reference one, and you kind of get the idea of what species you are looking at without like sometimes like when you sample from water or from soil, you don't even see the actual organisms. You just know that DNA is in there, it's been there at some point. So you can blast it easy. So this like require no understanding of molecular biology whatsoever. You just like, I got the ATCG, what is this? And you like Control C, Control V, and then hit blast, and like that's first thing, that's the right one. Yay! So that's just how lazy the taxonomists are when it comes to molecular biology. So the data sets are really good for the most part. For the most part, you have to keep in mind that this is come out of good view of scientists around the world putting data together, and some of them might be mistakes, not because they're not careful, but just sheer amount of data, and you're about to make some mistakes sometimes. And those two parts are easy. This is the hardest part for me. And I work in this area. I could be talking a bit too much about it, but amplifying PCR, I can't write PCR is, if you've done the lab, PCR is one sad little realization. It worked one day, the next day it wouldn't work. I don't know why. We do the same exact thing, probably like step different foot out of the house that day. Who knows? My postdoc having a nightmare right now because she have first successful PCR, and then yesterday she's like, it didn't work. It's like, it's actually the same thing, right? It's like, yeah, it worked yesterday, but it didn't work today. It's like, okay, so do it again. So that's the story. But that part is biochemistry. We're going to touch that. This is a difficult part. Where do we look in the whole genome? There's like the whole lot of like DNA is a long stretch of thing, right? And you could, in theory, you could look at possible regions to figure out what it is, but most of them would not be useful, right? You just need just one small region to tell. And the question is, which one? Which one would be the one that you want to look? You don't want to spend forever time figuring that out. So thank God there's people who think of this before us. So there's a way to decide whether or not some region of your DNA is a good one. First one have to be easy to amplify, in parentheses, across a range of species. So we want some region that easily PCR, like PCRable, if that's such a word. So like something that we can extract data out of quickly. If you've done any molecular lab at all, you feel this pain, like this thing worked in that bacteria and didn't work in that bacteria, and just like, and if you chemist, you spend your life try to make that happen. But then if you're a biologist, you want to get your work done quickly. So you want to work with the region that would be easy for all species. So this criteria is called universality. Is basically that it should be universal across different species you want to cover. If you want to study fish, like this marker should work with all fish. It should not just work with like catfish or salmon. It should work with everything on your sushi plate, basically. And they did use that. They did use this to catch the illegal fishing. So in the exporting, they claim it to. It's really fun because actually my friend did this project in Taiwan, and it's like, is this sushi salmon, as they say? Ooh, right? It's juicy. And it turned out half the restaurant that served salmon, it's actually they serve trout, not salmon. So shitty, right? So this allow you to do that. So first of all, your alignment, your barcode region have to be easy to amplify. And the second had to be easy to align. You already do the alignment. If you do that protein coding region is probably not that bad. And some of them have a lot of gaps. And if you click a line and it looks really weird, you should not use that. And remember, this is like systematics doing molecular biology. They don't want complicated story about, oh, we can fix that, we can do PCR better, we can sequencing better, like blah 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 blah. They won't want to hear that. They want something quick and easy. And most importantly, this is like at the core of the belief that region should be different between species. It should be able to tell the species apart. And this criteria has a name. It's called discriminatory power. So that's something that we must have, right? So I noticed that a lot of you are like in and out, must be the internet at Salaya. So I'll make sure I upload the video quickly today so that you can catch up. So, and this is what I'm gonna focus on. This is a discriminatory power. How do we decide how much power do you have in separating species? And there's two main ways: the distance-based measure and the tree-based measure. Just like before, just like when you tell the species apart, right? So there's a couple different names of tests that you can use. You can compare intra, meaning within, and inter mean between specific distance, and there's like base match and base close match method. I'll talk about that. And in the tree-based measure, we use monophyly of the species, just what we learned before, if the species monophyletic. So let's go through all this methodology. So if you use inter versus intragenetic distance, so what you do is you sequence all the samples. And if they are the same species, you measure their intra-specific distance. So the genetic distance between same species. And this should be pretty small, right? Because you're the same species, you should not be that different. So this should be pretty small. And then you measure another one is the inter-specific distance. So this is between species. So if your marker is really good, the distance they see between the species, between the sample of different species should be pretty large. Whereas the genetic distance between the same species should be pretty small. So the idea here is that if it's a good barcoding, for good barcode region, the inter must be larger than intra-specific one. Simple. So that region of DNA should have that properties. Not all the region have this property. Some of them just equally diverging for everyone. So you want to find a region that like within the species, they're pretty similar, but they look really different when you move to different species. Okay, so that's the idea. So this one example from one of my students' work. So you can see the inter is in green and intra is in purple, and the inter is in all. This is of three different markers. They're all bigger, right? So the inter is in all cases greater than intra-specific one. But then like which one is actually better if you have to pick one? So like, there must be another way to look at the ITS look pretty promising, right? Because this difference between inter and intra is quite big. So this is what we define as the barcode gap. The all barcoding gap is the difference between inter and intra. So if you have intra-specific minus intra, that's a gap. And you want the gap to be as big as possible. You want the difference to be really clear between the inter and intra. because when you pick two samples and you measure the differences, if different really large, and you can decide, oh, my difference is really large, it's like at 0.6, 0.06, then it must be of different species. If my um, difference is only 0.00, 0.01, it's really small, it could be um, um, in the inter-specific. And some people even go further. They're like, oh, that's not good enough. You cannot just use the mean. You have to be very, very conservative. When I say this is, all right, do this again for me. Oh, I can't share screen. Weird. Okay. Okay. So for some people, that's not good enough, right? You can just do mean inter minus mean intra. And I was like, yeah, that's probably good enough. But some people like, you know what? We need to be very conservative. We need to be like pretty sure that they're very good. So they're gonna use the smallest number here. They will give you like the minimum inter minus the maximum, like the highest of the intra. So like the smallest difference between the species must be bigger than the largest difference within the species. That's just how good it has to be. Some people want that, and it's sometimes so difficult to get. It's not like not all your DNA markers are that perfect. It's gonna be hard to use. And two was talking about use Euclidean or correlation distance, and as I said earlier, the way we calculate genetic distance are many, and you can do a lot of different distance. You can use Euclidean, you can use correlation, you can use all those different methodology. And for some reason, I don't know why. I think it has to do with legacy or tradition. The distance that used most often in barcoding region is Kimura 2 parameters. If you remember, this is when they give the different rate for transversion and transition. So if you move from two rings to two rings, it's one rate. And from one ring to one ring to two ring is another rate. So it's a different rate for transition and transversion. So that's how we use the distant bit method to uh, figure out whether or not that marker is good or bad, right? Another way is a bit more coarse. It's basically like you have reference here. This is reference of a species green, and this is the reference of species orange. And you pick some random sequence from the back, and it's like, which one is this matched to? And best match meaning like smallest genetic distance. If you ever paint your house, you do this. I know you've done this already. It's like you go with the color palette, right? It's like, and you go to the shop. It's like, is it that color? Is it that must be the color? And you do the same thing with genetics, right? It's like you go with your DNA. It's like, is it close to that one? And you're like, found a match. And like, oops, it's not that if that species. That's good. You get a crocodile butt, and you're like, where is it compared to? Oh, crocodile. Good, very good. And then the lower part here is like you do the same thing, and like I have crocodile, but it's a crocodile, and like it's a chicken. Like what? That is not right. So it did happen sometimes that like the closest match you have is not your own species, and that's when you fail. So the best match is they count the number of success. They do this multiple times with a lot of samples, and the more success you have, the better. So they use this as a way to tell whether or not that marker is successful at separating species. And finally, this is like the last the method that we already familiar with. I asked you like 5 million times what the monophyly is like, because if anything, you are on this class like cladistic people love monophyly. We want everything to be monophyly at all costs. So even if it means and you're gonna have most problem with mammalogists, because most of mammal species are not monophyletics, but then they try to keep it that way because mammals are mammals, you know. Anywho, so in this case, so this is from my own work, and these two trees come from the same set of species, same set of specimen, and you can see that only one species out of one, two, three, four, five, six, seven. One out of seven species are actually monophyletic. It's only 14%. Whereas like these three species perfectly in their own group. So it's 100%. So this come from the same plant, but different markers, different barcoding region. This marker perform much better already. They turn everything into monophyletic groups, and you say, oh, you use different set. No, this is the same plant. Like me and my student, we extract DNA ourselves. The DNA come from the same tube, and we separate them into different PCR, so we know for sure that it's the same thing, and it should, if they were to group together, they should group together. So you can use monophyly as a way to tell like how many species you recover as monophyletic species. And the choice of trees is up to you. If you feel like neighbor joining, go for it. You feel like maximum likelihood, go for it. You feel Bayesian, go for it. And nobody gonna tell you anything. I seen worse. I seen people done everything in this field. They just want to find a reason to justify their choice of markers, and they will go at great length to do that. So so that's the two main methods, the distance-based method and then the tree-based method. And for those of you who might be wondering, like, should not we know this already? And the answer is kind of yes. For animals and fungi and bacteria, these are kind of figured out. So for most animal, they use CO1, which is the mitochondria region to tell the species apart, and they also use CO1 to trace human origin back to Africa. So CO1 pretty widely used in animals, but then I think people start to realize that that's not perfect either. There's a group that CO1 doesn't work too. So, but CO1 working pretty well for most things. And the first paper that worked on it is actually used to study butterflies. So the origin come from the separating species of butterflies. But in fungi, I use ITS, which is the nucleus one. And the bacteria almost universally now, they use 16S, which is the ribosome coding region. Remember, bacteria doesn't have organelles, so you just have like ribosomes and DNA floating around. So they have 16S, which is a smaller subunit. The eukaryote have 18S. So these are pretty standard. And if you send out bacteria for barcoding, they would almost always ask for 16S. And the same thing fungi. In the database of fungi these days, they even have the database called RepSeq, which is the, as you see from the name, is a reference sequence. And this is a fun connection between last lecture and this one, is that the project I was involved in a couple of years ago, quite a few years ago actually, so that we sequence the type specimen sequence and put them into the RepSeq. So now you have the actual reference because the sequence, that DNA come from the type specimen. So I was working at the museum and like get the type specimen that we have and then send the sequence over. So that's like become the type sequence of the type specimen, which is pretty cool. So we actually now have the sequence that, go directly to the name. So that's at least for fungi for now. So, but there's a variation. There's people who hate these markers, like they don't work for us, and the ITS is really controversial because it doesn't always work, even in fungi. CO1 work pretty well, 16S pretty well, but then like you're missing one colors, right? It's like the group that Achanbir is working on, and that group is plant. So what happened to plants? Don't we have the answers? And the answer is we don't have the answers. So we don't know. We don't have that region. Bad news, guys. We don't have that region. Repeat it. We don't have the region. It's like, what? Are you serious? Like molecular biology has been around forever and people have figured out what's wrong with your plants. Come on, you can do better than this. Like, we're gonna show you the reason why. Remember, we talk about the three criteria for being good markers, right? You have to be a universal, right? Had to be easy to align. And third, they have to be have power to discriminate. So the short answer is not no single marker in plants can do all these three. They fail. They fail the test. It's kind of sad, right? So the first one is this. So the first one we focus on universality. So the colors are different group of plants. There's angiosperm, gymnosperm, the naked seed, and the cryptogams. Like, what is cryptogams? So crypto, again, there's a lot of crypto thing today mean hiding. Gams mean marriage. So marry in secret. They have sex in secret. They don't have flowers. They don't have like big structure that produce seeds. So yeah, cryptocurrency, yeah, Bitcoin, exactly the same thing. Yeah, so these are like the new thing. Like the cryptogram is like ferns and mosses and algae and all that stuff. So they compare the percent of sequence obtained from these plants using these different markers. So for things like RBCL here, the R, it's pretty good. Like you can get the sequence almost reliably at 100%. But when you look at this, the K, the MAT K, for gymnosperm and cryptograms, you only get only 30%. Meaning, if you brought me 100 mosses and asked me to get the MAT K, I only can get you 30% of the time. And reality, this is speaking from my experience. I get, you want to guess how many percent I get MAT K from bryophyte? Anyone? Zero. I get nothing. I've been working for more than 6 years now. I've been working for like almost 10 years trying to get MAT K from the freaking bryophyte. I got nothing. Nada. Zeros. Like nada. Like I was like, oh my God. Like people keep telling me MAT K is awesome. Like you should use this. It's really great. And like, okay, fine, I'll try. And then like I spent a lot of money trying to get MAT K to come out. They didn't. And okay, so fine. Like I tell all my students just forget all MAT K, and then I have one student who worked on flowering plants. He worked on um, yams, and it's like I want to try MAT K. I was like, okay, let's try. And on his first try, on his first day, he get all the sequences, all of them, 100% successful on flowering plants. Like what? The world is not fair. It's like it's not universal. It's like this is discrimination against cryptograms. Like you don't love moss, don't you? And oh god, it's awful. And the reason has to do with molecular biology. Is there's a secondary structure of MET-K region in bryophytes. See the MET-K region in bryophyte kind of close up, so it won't allow for primer to attach. So it's a difficult part to get to. So basically, this all popular plant marker, they mostly fail at universality. They don't apply across the board. They only work with a small group of plants. And the one that kind of make it are these P and R. So remember these two for now. Like P and R kind of work, right? They work with all plants. This should be good. And then you move on to the next one. And we look at the discrimination success. And you will find that even your best candidate P and R have the maximum success at 70% and less than 80%. That's your best shot already. So I see Catherine cringing. I was like, yeah, that's the feeling. So that's exactly like when I tell my like I have a like really old school colleagues, and she's like so, like she's traditional taxonomist, and she'd been doing morphology forever. She's like, can you use DNA to check if this right species? Like, yeah, you can. Like, how precise they are? Like, about 80%. So you tell me if I give you 10 plants, you get 8 of them correct and 2 of them wrong. Like, yeah, that's the best case. Like, what? And the worst case is what, like 20% correct? What the hell, man? So you try to sell this idea of barcoding to other people. The animal people have great success, fungi people have great success. Um, the bacteria people already use it as a way to tell the species. They don't even care about morphology anymore. So the plant people will be like, you want that too? I was like, nah, you're not gonna have that. Like, even your best shot is not perfect, not even close to it. It's like 80% is all right, but that's the best case. That's like the best you can hope for. So people like really frown upon, and I work on this topic, and I get a lot of like that look, like, oh, you're looking at barcoding a plant, huh? How's that working so far for you, right? It almost feel like you're in like Slytherin, you like working with Voldemort or something, like dark arts of systematics. Like, I think there's people who believe that barcoding will never work for plant ever, never ever. And in my view, is that, well, if you get 80% correct, at least is 8 out of 10. So it could work. It better than nothing. It's better than me watching looking at the whole thing the whole time. I don't want to do that. So I mean, there's a relative utility of this. And then sometimes, like we talked about earlier, sometimes you have plants that come in like speck. You know, like you don't have the whole plant. You don't have the flowers. You just have like one dust powder, and you need to know what it is, because sometimes it turned out to be starch or heroin. That's totally different things, right? So you must be able to know that. So I keep coming back to this example. Why? It's just stuck with me. But let's talk about some actual nicer example, more pleasing to look at. So this is a beautiful orchid named Hokoklasm. Is in Thai convert prime as 3 species, but in China there are quite a bit of diversity. They're quite a build, and because I mean, um, just like everything else in the world, the beautiful things get desired, wanted, needed, transported, smuggled. So these orchids smuggled quite heavily out of the forest and abroad, actually. And if you into orchids at all, orchid seeds are like tiny dust. It's like dust. So I have a friend. He's an orchid enthusiast. There's people like that, and they're pretty interesting people. So this is when I was in Berkeley in California, and he's like, come to me, he's like, I got the seed from Thailand. I was like, what? I got the orchid seed from Thailand. I was like, okay, why are you telling me this? It's like, oh, you're from Thailand? Like, yes, but should I care about your orchid seed from Thailand? He was like, oh, it's like he probably anticipated me to be more excited about his orchid seed from Thailand, and he opened the package and it's like, what is this? I was like, it's orchid seed from Thailand. It's like, it's a dust. It's like a powdered dust. Orchid seed is smallest seed probably on earth. It's like so tiny, and doesn't have any endosperm, have anything. It's just like a dust. And this is how people smuggle orchids, not with the flowers, not with the plants, with the seeds. And you will have no idea, you have no way of telling what species they smuggle, because it's just fine dust. So you can tell me the barcoding sucks, but hello, how you gonna identify this powder of orchids? Good luck with that, right? So here comes barcoding. So this is a group that they're concerned in China, and they do the barcoding studies, trying different markers to see which one works for them. So let's walk through the result together. So they have 5 markers here, right? And they said that the universality of primers being meaning how much success they have a PCR. So the first 2 lines are pretty much the same properties, and If 100% is yes, it's universal. 92% is still yes, pretty good. Yes, yes, and then the p-cells is pretty low, so it's not universal. So this is now come to the lesson of what not to do. So even if your result is low, tell me the number so I decide for myself which one is low, right? That low might be 1%, that low might be like 89%. Like I don't know. Like give me the number, I decide whether or not it's universal or not. But anyhow, they tell us low. Like how many percent is that low? Like how many low? It's just like one of those people who just keep giving you the same answer. So they kind of drop this for further analysis. They're just like, you know what, it's not worth our time. Let's not looking at that. So they're looking at then the inter and intra specific distance, right? And they asking whether or not the inter were greater than intra, right? Because they have to um, look at a difference. So in all cases, right, they have bigger inter than intra, bigger number almost always. So that's good. So what happened to BCL? Like what's going on here? It's like no data whatsoever. So what do we do with what's going on? So you can see here number of indel. This is insertion and deletion. So there's zero insertion or deletion whatsoever. And you look at the number of variable sites. So this is number of difference you have in sequence is 8 out of 1200. So of all the species, all 12 species, there are only 8 differences among them. So the intra specific here is like so low, like it's zero essentially. Like if you are the same species, you'd have the same RBCL, just like zero. Like can't even compute. It's just like cannot compute. You guys too much alike. I can't compute. It's just like so weird. So they kind of dropped. I would have liked them to show the data, but they just decided it's not worth the time to go further. It's just like so different, which I don't understand why they don't include the data. But I suspect that's a reason. And then they look at this ability to discriminate. And this you see NJ and BI, this is tree based. Using the monophyly data to look at, right? So you can see that most of them are not perfect, and they like majority of them only be able to tell 5 species apart, and the other kind of mix up. So the best case here is eight species of 12, and that's belong to MetK. See, MetK again, like everyone keep telling me MetK is awesome, but then just like it does not work for me at all. So this is how you tell how they choose and look at all this different data, and different criteria, different tests might point to different directions, and you kind of have to figure out what's best for you. And sometimes they show it like this. They show the distribution of distance. So this is the number of pairs, right? The number of pairs that, and the black one is intra. So like when you read here, there's an 8 pair of samples of the same species. There's 8 pair of these that have distance of 1. Oh God, I keep doing this to me. Oops, where is my slide? Slide. Okay. So you can see here that it's another way of visualizing the result. So if you look at the intra-specific, they're pretty small in all cases compared to the inter. Oops, I should use different colors. The inter is a lot bigger, but you can see that for MATK, which is A, the difference pretty clear, and same with the ITS. Whereas for the last one, the third HPSBA, the difference is so small, it's almost overlapping. That between intra is not that big the difference. So this one is not good for barcoding, right? So they picked MATK for this study because pretty clear difference between the inter and intra. So that's an easy case. That's a nice, cool, clear case. And you like, is that nice already? It's only like 4 to 8 species of 12. Like, yeah, that's nice already. Let me tell you the horror story next. So this is a horror story from my own lab. This is mosses, sphagnum. So sphagnum is pretty typical mosses um, in um, in the temperate zone. Is they form the big bogs, like some big, and it holds so much water, the water become acid, and sometimes if you fall into this sphagnum box, you preserve. There's like a sphagnum man, like people who fall into the box and preserve, just like mummified in the sphagnum box. So yeah, don't get me mad. Oops, I did not treat anyone. So don't put, if you want to kill me, put me in a sphagnum box so I can be preserved. Nice. I should look up. I should have brought the pictures of sphagnum man. Is that such a thing? Oh yeah, this could look gruesome. So this guy, gonna sharing the screen, sharing a little bit. So this is the body that preserved in the sphagnum moss. So this guy fell into the box a couple 100 years ago, and it's perfectly preserved. Look at that. Look at that. Like how well preserved these guys are in the. But anywho, so sphagnum moss is being used for a lot of things. They are used as female napkins for the Native Americans, and yeah, because they absorb a lot of water. And but the main thing is that they're so difficult to identify. So there's a 5 species of them. When you put them side by side, they look kind of different. But like for me and my student, they like look really similar to each other. It's like, huh, like okay, I can see difference between 1 and 5, but then like 3 and 4, like like you have like crazy headed and like not so crazy headed. That's it. That like, and 1 and 2, like you skinny one, you fat one, like so it's like really really tiny differences, and we want to see like. And when I walk into this project, I'm like so confident that like, my DNA barcode is gonna work. Like, gonna prove them wrong, and like we can use it in plants, and this will work. I don't have to bother with this crazy morphology anymore. And then, oh, oh, it's like, oh, what? Like, there's 3 clades, but then this has 2 species in there. This one also have 2 species in there, and they all kind of mix up with each other, so they're not monophyletic. And only species C is come out. So my success rate is I have 5 species and only one works. So that's 20% success. Yay. And this is like my student, his bachelor thesis. He's like, oh, he's like really excited. He got his work done in his 3rd year. Like he finished all the molecular work by 3rd year. It's like, oh, so it doesn't work. So like, okay, calm down. It's probably not what we think. It definitely not working the way we want it to, but what's going on? So like we dig deeper. So I make him do the phonetics, the morphology using PCA, and we do the coloring by the species based on morphology. So these dots here based on the morphological data, if they're closer together, they are similar morphologically. And if they're separate, they're different. So species C here, the SC, is the same one as clade C here. So this one is easy. They're so different from other species. So it's pretty clear. If you look at the pictures, it's this thing, is the skinny looking one. So it's clearly different from the others. So that one is fine. They're like on their own. And then you look at these two pairs, like they're almost overlapping. They're like very similar to each other. And it's like, okay, so probably not our fault that we can't tell them apart. They're like really similar morphologically. And let me tell you, this come from the character matrix of over 100 characters. He spent like another half year scoring this, like looking at every specimen, like 0 1 0 1 0 1 0 1, and he's probably gone crazy. And yeah, he now working on immunology. He doesn't want to do taxonomy anymore. So it's like that's probably why. And we look at it again, it's like, okay, so that's different. So we use the same dot. The dot is at the same place. You can see here it's like at the same place. But now we're grouping by the genetic group. So this is by clade. So this is by the ABC clade here. And you can see that, the A clade here is actually kind of grouping together nicely morphologically too. But it's not monophyletic, so what's going on? So the question is, so is the species delimitation correct at the first place? If like you separate SJ from SK, right? Using morphology alone, and there's like some differences, but when we look at genetics, they're kind of similar to each other. So they're not that different. They're not that different, and they're probably um, in the same group. So maybe, and I just say this because we haven't dug into or tried methods to say it, but we we think that it might be the fact that the taxonomists are splitters. They're splitting things too finely. They try to find a different when there is none. It's just very one variable species. And we found this trace in the literature. We found that these two species used to be subspecies from another species before, and then they now get separate into their own species, even though genetically speaking, oops, sorry, yeah, you seen that uh, picture before, even though genetically speaking, they're not, uh, oops, sorry, I had to do. So even though genetically speaking, they're actually the same species, there's no difference between these two species that they try so hard to divide it using just morphology alone. So, and when I say morphology, they use a few key taxonomic characters to separate them, but in genetic wise, they're not that different. So we have pretty good reason to show that these two species are probably preferably might be just one big groups by genetics. And it kind of go back to how we draw the line. Like, is it the right line to draw to begin with? So I guess if anything to walk away from this is that no method is perfect, right? We always find a way to make our life easier, a way to have multiple evidence to figure out what the species are. But at the end, biology is biology. They the sphagnum doesn't know they want to be separated to different species. It's just like I'm hanging out here waiting for the dead man to fall in. So you know, so there's a place that barcoding works quite well, and there's a place that barcoding fail, like mine. So, and I'm trying to figure out a way to write a paper about this. So, like how to fail at barcoding. I should say that. Oh, that's a new title of paper. So, how to fail badly at barcoding, just like rub it in people's face. So that's kind of moral story that DNA coding is a new wave, and people are really excited because finally they don't have to take taxonomy class anymore. Just learn molecular biology and just sequence DNA of everything. But there's a lot remain to be done, and as long as the species line are still not clear, barcoding is just like bad tools for the wrong job. So barcoding is useful when you have very little to nothing to work with, and there's a last resource you need to, and you probably get to the right genus, probably not the right species, but that's probably good enough for most things. So that's the gist of it, and